Good morning, everyone, and thank you for joining us. I am Amanda Ritchie Terzian, Mrs. Syracuse 2021. I am proud to also serve as the STEM Goes Red Committee Chairwoman for this year's Go Red for Women campaign. Before we start, one quick note on the best way to participate. We encourage you to enter comments and questions into the chat box. Our researchers will be answering your questions during a Q&A portion later on. Make sure you put them in the chat box and we'll save them. But before you do, please select all participants and attendees from the drop-down so all those viewing can see them. Now, let's get to our inspiring program. Go Red for Women is dedicated to removing the barriers women face to experiencing good health care and well-being. The price of being a woman is high, and when it comes to cardiovascular disease, 1 in 3 women pay that price with their lives. The work of Go Red for Women is more important now than ever. We all know this pandemic has been a stressful time, but it's been even harder on women. More women have been out of work than men, and for those women who are working, they are taking more of the lead, balancing virtual learning for their children and their jobs. But together, we can make an impact on women's health. We can protect our mothers, sisters, daughters, and friends. Go Red for Women is also dedicated to removing the barriers women face in the realms of health, research, and science. The reality is, our health is about even more than our bodies. We know that lower levels of education are associated with a higher prevalence of cardiovascular risk factors, higher incidence of cardiovascular events, and higher cardiovascular deaths. Removing barriers to good health and well-being includes closing gender gaps in research and in STEM fields. Black, brown, and multiracial women, including those who identify as female, continue to be grossly underrepresented in cardiovascular research and STEM careers. Research shows women experiencing cardiac events potentially have better outcomes when treated by a female physician in the ER. We need more women creating solutions for women. It wasn't until 1993 that women were required to be included in clinical trials. Even now, only 38% of cardiovascular research participants are women, and women of color are significantly less represented. Women make up nearly 50% of the workforce, but less than 25% of STEM careers. In fact, only about 3 out of every 100 female STEM degree holders will go on to actually work in a STEM field. In this context, the American Heart Association is proud to present today's conversation with 2 local female AHA-funded researchers as part of this year's STEM Has Heart initiative. This incredible program wouldn't be possible without our national sponsor, CVS Health, and our local STEM Has Heart sponsor, National Grid, plus additional local STEM sponsors, St. Joseph's Health, Nescentia Health, and United Radio. Now, it's my pleasure to introduce our 2 featured presenters. Dr. Plansky Hong is a graduate of Henninger High School. While studying at Syracuse University's College of Engineering and Computer Science, her research received funding from the American Heart Association and was awarded a highly competitive and prestigious predoctoral fellowship. Her AHA-funded subject, the effects of drugs on the heart health of developing embryos when taken by pregnant women. After earning her PhD, she remained at SU as a postdoctoral fellow. Hong is now a researcher in the Syracuse Biomaterials Institute, working in the System Tissues Engineering and Morphogenesis STEM lab under Assistant Professor Zhen Ma, and in the Henderson lab under Associate Professor James H. Henderson. She has been developing an in vitro model that tests various medications on developing heart cells to contribute to the prevention of congenital heart defects. Her work helps determine which drugs, when taken by pregnant women, may be harmful to the heart health of developing embryos. She evaluates the effects of drugs that treat everything from acne medication to cancer treatments. We'll first start with a visit to Plansky's research lab.
Hello, everyone. Thank you, Amanda, for that very kind introduction. I'm really excited to show off our research lab today. So I work in the Syracuse Biomaterials Institute, as you can see behind me. And what the Institute is that it's a collective of approximately 7 faculty who range in disciplines from tissue engineering to material science and chemistry. And the goal of the Institute is to bridge the gap between biomaterials and that of biology. So we want to try to understand how we can engineer materials to change biology or to manipulate biology. So let's go ahead and get started with the research tour. It'd be much better to do this in person, but because of the pandemic, this is the best we can do. And I hope I can thoroughly show you everything using this format. So as we go in, we have the sliding doors to the main chemistry lab. And this is a large area where people perform chemistry reactions in order to make different materials. So we make materials that are suitable for different biology applications, and each research lab does different work. We have one lab that focuses on 3-dimensional bioprinting. We have another lab that focuses on shape memory polymers for wound repair, and so on and so forth. So once we make the materials, I'm heading over to this area here where the materials are further characterized. So in this area here, we have different testing instruments so that we can test the properties of these materials that we're looking at. And furthermore, we have things like rheometers, which test material viscosity, material strength, and so on and so forth. So now I'll take you to where I primarily work. As Amanda mentioned, I work a lot with in vitro tissue modeling. And what that means is I essentially culture tissue. I grow cells and I grow tissue in a dish, and I see how they respond to different things like drugs or chemicals. And we have this separate room that is for cell culture and molecular biology. And this is where all of our cell work is formed. So when we make the material, we characterize the material, and now we grow cells on them. And in this room here, so this room here houses numerous incubators where the cells are grown and cultured, various equipment for proper cell handling and analysis. And in this room here, I'm showing you guys the to analyze them. So from what I've shown so far, it's essentially we start with materials, we characterize materials, and then we see the biological interaction that happens on these said materials. So now I'm taking you to another room where, so there are a lot of people in there, so I don't want to interrupt anyone's work right now. So it is pretty busy. It's the peak of summertime where we have a lot of summer students coming in. And We're hosting different research experience programs for undergrad students. So there's a lot of training, there's a lot of talking going around. So, and we're really happy to host these programs every summer because it does bring in a flood of new students to learn what we have to offer here. And a lot of these students actually come back for graduate school when they're finished with their baccalaureate degree. And here is just another room where we analyze biological materials, or where we analyze biological samples. So this is a PCR machine. This is our essential PCR area. And then we also have another lab that works with bacteria. So what they do is they look at the antimicrobial activities of honey on these materials, and they see how these compounds in honey kill bacteria for more safe and sterile wound dressing. So if you get a cut and you have this special Band-Aid that you can put over your cut, it will help protect against infection. So that is the gist of the full lab. And one of the things I really like most about this institute is the collaboration. So we do have, so as I mentioned earlier, it's a collective of approximately 7 different faculty that range in disciplines. And because everyone is there's no one section for any one professor. So there's a lot of collaboration going around, and we can learn from other labs, learn different techniques, and learn different expertises in order to advance scientific research farther. So I think that's all I really had to show. I would love to take questions at the end. So I'll hand it back over to Amanda.
Thank you so much, Plansky. You're doing amazing work. That was so neat to see, and everyone start thinking of your questions while we move down Route 81 to spend some time with Melissa. Melissa Mendoza is a PhD candidate and an AHA predoctoral fellow. Her AHA-funded subject, 3-dimensional valve-on-a-chip models of the aortic valve fibrosia. After living in Queens, New York, and graduating from Rochester Institute of Technology, Mendoza is now in her 4th year at Binghamton University, getting her PhD in biomedical engineering under Dr. Gretchen Mahler in the Organs on a Chip laboratory. Her graduate research focuses on developing a disease model to understand the onset and progression of calcific aortic valve disease, in which the only current treatment is total valve replacement. With this model, her research hopes to play a role in the development of new drug treatments, limiting the number of animal studies, and providing more effective clinical trials. Mendoza credits her mentor and professor, Dr. Gretchen Mahler, with leading her to advance her career in science and research. Mendoza pays this forward by serving as a mentor to undergraduate students and encouraging their work as they go into scientific research. And when asked, where do you see yourself in 20 years? Her reply, I want to be a working woman doing innovative research and sharing my knowledge with others. Melissa, welcome.
Hi, everybody. Thank you, Amanda, for that lovely introduction. Sorry, one moment. Okay, so I am really happy that you've joined us here in the lab today. I am excited to show you around. And so right now I'm located in Vestal, New York, and Binghamton University's Biotechnology building. And specifically, I'm in the Organs on a Chip lab. So here I work under Dr. Gretchen Mahler, And our lab consists of a variety of students from all over the world, which is really exciting. We have one postdoc student, 8 PhD students, 2 master students, and 2 summer undergraduates. As Clancy mentioned, the summer brings a lot more people around to do exciting research. So to me, we're a pretty large lab, so it can get a little bit hectic at times, but what I really enjoy about that is that we all work on so many different kinds of projects related to all different types of organs. So we look at the heart, we look at the kidneys, and we look at the gut microbiome, specifically the small intestine. So there's a lot of exciting work going on in this one lab, as well as in Binghamton in general. But today I'm actually standing in one of the 3 lab spaces that compose our entire organs on a chip lab. So let me give you a brief view of what the room looks like that I'm standing in today. So this is one of our cell culture rooms, as Clancy mentioned, some of the work that she does as well. And just a little bit about my work. So my work falls under the realm of microfluidic models of disease. And yes, these are really complex words that, to be honest, I didn't even understand a couple of years ago. But what this means is that we use really, really small tools to apply really small amounts of liquid to biologics. So what that means is, cells, proteins, growth factors, etc. And we use all of this to create disease here in the lab. So this has many applications. As Amanda mentioned in my introduction, not only does it help us to understand how disease might begin or continue to progress, but it could also help us to potentially find ways to treat them. Leading to more effective clinical trials and the need for less extensive animal trials even. So let me show you an example of a device that I use. So it's a little clear, it's a little hard to see, but it's made up of materials such as silicone and glass. And these models we use to embed cells that we actually isolate from the aortic valves of pigs. And what we do is we take these devices and hook them up to pumps that continuously allow flow to pass through them. And so this recirculates things like nutrients and growth factors and proteins, for example, just like the blood passing through our valve in our hearts. So I actually have an experiment set up today to show you what that might look like here in the lab. Let's see. So this is called a peristaltic pump. And as of now, I have 4 devices hooked up simultaneously. And over the course of the next few days, these models with cells and everything inside will begin to exhibit calcific aortic valve disease. So this fancy box I'm showing you here is called an incubator, and Plansky showed some of those in her lab as well. But what's really awesome about these is that they help to regulate the temperature, the humidity, and even the gas exchange that happens to allow our cells to grow and flourish and do what they need to do inside. Another thing that we use a lot is a biosafety cabinet, as seen over there. And what's really great about these is that they allow continuous airflow and filtration of that air. And this is important because everything that we do here in the lab needs to be maintained sterile. And what that means is that we don't want to introduce contaminants into the work that we're doing, whether it be like bacteria or fungus, and that may affect our experiment, but also we want to keep ourselves safe. So whatever it is that we're working with, we also don't get hurt by those things that we're working with. So I guess one of my favorite things about working in this lab or doing research in general is that I get to develop tools and investigate them and things that haven't been done yet. And I know that can be really challenging and kind of scary at times. But it's really, really awesome because when you hit that moment that like things like actually work, you really realize that it makes all the difference, and you really learn to appreciate the hard work that you put into your project. So my day-to-day life, I'm not always in this room doing cell culture and things like that. But yeah, running experiments, taking care of my cells and making sure I'm prepared for everything that I do, but also working on things like data analysis, or even making presentations and writing papers. So research keeps us pretty busy, but it's also really exciting work. So with that, that's all I had to show you today, but I look forward to taking your questions, and thanks again for virtually visiting me here in the lab.
Melissa, thank you so much. Your work is interesting, and on a personal note, my father had to have aortic valve replacement, so I appreciate the work that you're doing to continue to help our world. So let's bring the two of you ladies together, and I have a question for both of you, and we'll start with Dr. Hong. As women entering the research field, how have female mentors been key to you being where you are?
I actually never had a female mentor, at least in research. But I have, like, throughout college, I did, there were females around for me to seek career advice from, and they've always been there. But I think the most key women who've helped me build my work ethic and my passion for science are people in my family. I'm the youngest of 7 children, so I have 3 older sisters. One of my sisters is a physician, so she's been extremely supportive of my career as a PhD student and as a PhD. And all the women in my family have extraordinary work ethic. My parents are immigrants, so I come from an immigrant family. So just learning that work ethic has been key to how I view life and how I tackle on challenges.
Melissa, what about you? You mentioned Dr. Gretchen Mahler, I believe.
Yes, so she's my my boss right now, and she's been really great. I think what I would say about mentors in general is that I'm just grateful for the opportunities that they give you. When you don't have the experience, or you've never done research before, they're always so willing to teach someone that's so enthusiastic about learning. And so like I definitely learned that from a lot of my mentors. From women mentors specifically, I think I've had a different viewpoint on like the work-life balance. Because it's something that we always dabble in and try to talk about, but it's kind of taboo. But all the women mentors that I've had have families with younger children, and it's really inspiring to see them be able to take care of their family, but also be like hardworking women.
Absolutely. Representation is so important. Absolutely. Well, my next question, and Melissa, I'll start with you this time. As researchers, what has been your greatest obstacle to date?
So like I mentioned in my little spiel, like challenges arise all the time. I think the most pressing obstacle is like failure. You do an experiment so many times over and over again, and then you finally figure out, that's the one thing that I needed to do to make it work. And you just have to learn from those mistakes. You have to learn from what's at hand all the time, and everything that you fail, it's not just like, okay, this is one negative thing happening. Like there's little positive victories along the way, so learning from those victories.
And Plansky, what about you? What are some obstacles that you've overcome or faced?
Yeah, so I agree with Melissa that failure is one thing, and it often leads to, especially like when I talk to other women in my discipline, my colleagues and stuff, A lot of us have this whole imposter syndrome kind of thing where, we feel like we don't belong sometimes because things fail, and like we're all kind of like perfectionists, especially in research. We want everything to work out well. So I think the best thing to overcome these obstacles about imposter syndrome and things like that is to just find a good group of friends, find friends who have the same interests as you. And then you can talk about your failures and laugh at them a week later about why they happen, because they do happen and they are learning experiences. So the sooner we can accept that, failure is a learning experience, like I feel like that's like the better off we will be. And, that way we can move forward in a much more smooth and progressive way, as opposed to beating ourselves down all the time.
Well, thank you both. We're going to turn to our attendees now to join in on the conversation. So to those who are viewing, please enter any questions you may have for these two inspiring leaders in research into the chat box. And while you're taking your time to think about your questions and typing them in, I wanted to share a little bit more with you about what the AHA is doing. So, did you know that the AHA is currently funding 8 projects, and 4 of those are led by women, with more than $804,000 right here in Central New York and the Southern Tier? Well, the AHA can use all the help it can get to help keep research such as Plansky's and Melissa's funded. So here's how you can help. Please consider taking a moment to send a donation to the AHA by texting the words "Go Red SYR" to 41-444. Yes, just follow the prompts. And again, I'll repeat that for you. Open your heart and text the words "Go Red SYR" to 41-444. I'm going to take a moment, pause, let you take out your phones and do that right now. Every dollar allows the AHA to continue to be a leader for women in health, in science, in research, all in the mission of being a relentless force for longer and healthier lives. One more time, Go Red SYR to 41-444. Now, let's turn to our viewers and attendees and see what questions they have.
Our first question is, it seems at first your work is individual. So how does collaboration support your goals?
Dr. Hong, why don't you take that question first?
Yeah, so it does seem individual at first, but we do, sometimes the individual work leads to collaborations with other work. So like a lot of my work deals with, I use a lot of different engineering techniques, but then I also use a lot of biology techniques as well. So so one of the collaborations I was able to work on, so I do the tissue modeling here that our individual lab does, but then we collaborate with a lab down at Upstate Medical University to compare animal studies as well. So that's one collaboration. And also with the different techniques that we use, other labs also kind of see what we can do and see how our techniques can apply into their techniques and vice versa. So all of our equipment within the lab, it's all shared. So anyone has access to use whatever they want for whatever purpose. And this is how we exchange different ideas and different projects and move forward with our research areas.
Yeah, so
Melissa?
yeah.
Oh, go ahead. Yeah.
So yeah, it seems individual in the beginning because you're trying to draft up a project that is a long-term project. A PhD is in a long haul, but yeah, a lot of the work that we do is collaborative. Like for example, my project is built between a collaboration of the biomedical engineering program as well as professors in the mechanical engineering department. because a lot of the work I do isn't just biologics. It's not just biomedical applications, but also, as I showed you, we use like pumps and all these kinds of things that is very mechanical heavy. So learning from those two different types of disciplines, but also like within our department, we do a lot of collaborations. I've been approached before to, work on projects that are not even related to my main project, but I've been able to contribute and publish and help out a different lab with their cell culture. So there's always opportunities to collaborate like within different types of labs.
Our next question, what do you see when you look at where your research will impact future research? How does your research build the next building block or the next discovery?
Dr. Hong, would you like to begin?
Yeah, I'll go ahead. That's a big question that we always get as researchers. Like, where do you see your research going next? Like, when you're presenting your work to other people, they always ask, where do you see your work going next? But one of the things, like, it's hard to see where it goes right away. There's not always a clear direction at first. But what we always try to keep in mind is the bigger picture. So, how can, what's the bigger picture of our research? So my work, like I like to describe it as a way to expand drug screening methods in clinical work in clinical areas. And although it might in the future, it might get the model that I use, it might get more refined, it might get tuned a little bit. At the end of the day, it's the same work. And when you publish research, and what you do is you give other researchers different ideas of where to follow up with your work. So it's more of like, publishing, throwing out that knowledge out there, and to just see what other people have to see and say about it, and how they can also expand the work as well.
Thank you. Melissa?
Yeah, so we're kind of developing, these devices, and I'm showing you like how exciting all this work is. And but you have to remember that it's still like early development phases. So we're doing really exciting work right now. And, I've been my 4th year now trying to work on this project. But, when you submit grants and you write papers and such, as Plansky mentioned, like this is just one stepping stone to like the larger picture. And so, this device could then further be developed by, the next student coming in and the next student after that. And hopefully, we could take it to, test drugs and such. So this is just like one small piece of like a huge puzzle that we're working towards. But it'll be really exciting to see when it does reach that level that you were able to contribute to that.
That's
sticking with the topic of your research and discoveries, what was your most exciting discovery to date?
Melissa, would you like to start?
Sure. So yeah, like I mentioned, it's been 4 years in the making. But I think my most exciting part was actually pretty recent, post-pandemic, which is also really exciting because you've been kind of out of it for a little, and you come back and you're trying completely new things. So I started a new like imaging technique to really look at the microstructure of what might be happening inside these devices. And we were able to show that we're actually developing mineralizations and calcification at the micro level inside of these devices in the lab. So to me, that was like my big breaking point. And we're really excited to publish this really soon. So yeah.
Plansky, what about you?
Well, one of the things I love learning about is like, they're not necessarily discoveries, but there's always new techniques and there's always new equipment coming out. And it allows us to discover new things within our system. So for instance, a really big technique known as single cell RNA sequencing has been used, and we've been implementing our model in that using that technology. And what it does is it allows us to see what kind of cells are developing in our organoids, our miniature heart model. So we've discovered that, you know, a lot of them, they're not only cardiomyocytes or heart beating cells that are forming, but we have a collective of multiple cells that all act in coherence with one another. So one cell supports the development of another cell type. And like I said, in our organs aren't just one cell type; there are multiple cell types. So, learning how closely we can resemble a heart is it's what's been really exciting lately, because we can see more how closely they resemble a heart and resemble biology, and this just opens new doors to how we can fine-tune medicine and how we can use these tools to predict medicine as well.
We have another question. What was the watershed moment that inspired you to go into research as a profession?
Plansky, would you like to start?
Yeah, I'll go. So yeah, in college, I didn't even want to go to research. I didn't want to pursue research at all. You know, it's just, I just had the goal of getting a job and calling it a day and, you know, moving on with my life. But, you know, one of the professors here who I worked with, he actually encouraged me to pursue research. You know, one of the things I didn't know is that PhD programs pay you to do research. So that was, you know, that's a great incentive to do research. But, you know, what he inspired me to do was that, you know, research is bigger than, you know, working for a company or just, you know, simply making money. So, you know, I decided to apply for grad school, and turns out I really liked it. I liked learning new things. You know, I like the flexibility of being in research and just simply just sitting around learning, not sitting around, like I'm obviously doing work, but you're always learning something new, and there's always something new that comes up that inspires you to do more or to work farther in the goal.
Melissa?
Yeah, so I started research in undergraduate years, and I think what really inspired me to like keep going was my mentor that just kind of told me, hey, like, you know, you're doing this work, like, why don't you go to like this conference and present your work and do all these things? And I was like going to conferences, you know, meeting people, networking, like learning a lot about all the work that can be done in research and how exciting it all is. And I was like, okay, like, let's go to grad school. Like, I just want to continue to learn, as Plinsky said, and just like discover new things and be a part of like, you know, trying to find treatments for diseases and such. So, you know, really just getting my work out there and talking to people and how excited like they get when you're excited about your own work.
What advice do you have for people who are starting out in STEM careers or looking ahead to STEM careers?
Melissa, why don't you start?
Sure. For those starting, I think it's just about starting with a conversation. Like if you know, you see us here talking about our research and you want to reach out and talk about the work that we do and how to get involved. I always tell students, so, you know, and undergraduates that if they want to do something, they have to ask. They have to approach, you know, the professor and approach the students and talk to them, because that passion and that drive that they show is what's really going to take them far. So starting those conversations, or even in younger years, like if you have like younger kids at home that may be interested in STEM, like do small like craft projects with them related to, you know, science, and see how they react. But yeah.
Thank you. And Dr. Hong?
Yeah, to follow up on that, yeah, it's really important for, especially if you're at the undergraduate level, to reach out to people, especially if you're interested in research. So, and if you want to go to grad school. And that helps build connections. My advisor, Dr. Ma, he's exceptional at getting names of his undergraduate students out there to other professors, and he's able to. He has a large network of other professors that he also communicates with regularly. So it's just good to get to know if you do get into a lab, work hard at it, commit to it, and if it's not for you, then it's best to move on, because the research isn't for everybody. The environment, it's a lot different than working a 9-to-5 job. But if you do enjoy the research, commit to it. Make sure you're communicating with your PI a lot, and there's this whole different relationship when you're a graduate student with your PI as opposed with your advisor as opposed to an undergrad. It's more of a colleague relationship as opposed to like this mentor-mentee relationship. So you become very close with your advisor, and to the point where like it's almost like a friendship where they share things with you and you're able to share things back. So just building that relationship will help just push your career ahead.
Thank you, ladies, so much for your insight, your valuable information, and just sharing all of this really interesting information with all of us. And with that, we are done with the question and answer session. And I just wanted to say, as we wrap up, through the STEM Has Heart program, we hope to continue to inspire and support more women like Plansky and Melissa. If anyone would like to get involved with the STEM Has Heart program, please reach out to Trina Tardoni or Kristen Thompson Henry at the American Heart Association. Their emails are on the screen. I'll give you a moment to take those emails down. Thank you again to our inspiring leaders, Plansky Hong and Melissa Mendoza. Thank you all for joining us, and I hope you have a great day.